there's one thing that's interesting that they found, is that even though the pancreatic enzymes couldn't break it down and the brush border enzymes here couldn't break it down, there's an enzyme produced by bacteria used in the manufacture of Swiss cheese that would break it down. And I mentioned this to a colleague of mine, a Swiss doctor who actually lives near the Emmental region where they produce Swiss cheese. And he said, you know, it's interesting. When I eat bread, I have a problem. When I put Swiss cheese on it, I don't have a problem. So it's very subtle things going on a molecular basis that are just beginning to uncover. Years ago, I wrote a book about this. I'm going to sign copies for anyone who's interested afterwards. We've seen a number of studies showing that taking reactive foods out of the diet results in a lot of improvement in different conditions. We have a group of patients here with dramatic improvement in skin conditions. We'll skip over this because it's too complicated for the moment. We see more than half the patients being improved who have GI conditions by just avoiding foods that are deemed to be problematical. We see actually the research showing that the precursor of serotonin is consumed, is used as a signaling molecule inside the gut. We discussed that before. We see that actually, and this is how it came about that I was asked to write the book. We supported our lab did, at Baylor Medical College Sports Medicine and Performance Institute, looking at weight loss and body composition. And all we did is test blood and find out which foods activated the cells, which induced the response. Randomized the groups, did the test on everybody, 50 and 50. They were matched up for age and symptoms and so forth, and tested their body composition through underwater displacement technology prior to dietary intervention, and measured their scale weight and had them complete an inventory about their symptoms. And essentially, 98% of the group not following a calorie restriction diet actually lost weight and or improved their body composition, whereas the majority of the people who followed calorie restriction actually got fatter. And here we go. It's not the simple math of calories in, calories out. There's complex processes that go on once the foods go in, and what happens to it and how we metabolize it. Do we burn it as energy or not? And the blue bars indicate response to an array of symptoms that the experimental group had improvement in. And basically they improved in all areas except for the amount of food that they ate, because the other group was asked to restrict the calories. It may be a little bit difficult to see, but the categories are things that are associated with psychological states, secondary to food intolerance, and inflammatory problems like arthritis and irritable bowel and respiratory disease and gastrointestinal upset and so forth. Same thing was seen in a clinic, an outcome of people following an elimination diet with various typical symptoms associated with food intolerance, ulcerative colitis, skin, urticaria skin, and eczema being skin issues, perennial rhinitis, and so forth. And the outcomes, again, showing very good benefit. Actually, much more benefit than is typically seen against a wider array of problems, because they have a common source in one factor, inflammation. Then we typically see with specific pharmacotherapy, which is encouraging. Again, it reinforces what Hippocrates and Lucretius said. Food should be thy medicine, and medicine thy food. But for one man, it may be medicine, and for another person, the same food might be poison. So we're individuals. we've seen over the years clinical validation of testing for food intolerance. We also, in this study, we supported it with laboratory work, actually not with funds, and it was very independently run, at the University of London. And it actually was designed like a drug study with double-blinded, placebo-controlled oral challenges. So essentially, this would, to a scientist, demonstrate that what we see in terms of improved clinical outcomes is scientifically valid and not as a result of chance or as an effect of placebo. Just quickly a little deeper into the differences between allergy and intolerance. In the 20s, just a few slides on the history, two allergists wanting to find out what causes allergy, here were their names, took the serum from Prauslins who had an allergic reaction to fish, and they injected it into his non-allergic partner, Kuster, and then they skin tested him on that spot, and he then began to show a reactive skin response, which indicated that whatever causes allergies in the skin, because no one really knew. They had the reagent, but now they know it resides in the serum. In '67, they found out that it was an antibody. And it's this classical pathway, the antibody. I don't think we'll get too much into this, but if anyone wants to have my slides, I'm not sure if you have them in proceedings, but you can get them from me. Basically, allergy is a complex process that involves the uptake of the allergen into an antigen-presenting cell. It gets processed, it gets expressed on its surface, and it gets picked up only by a T cell that has a conforming receptor that clones itself, sends signals to B cells with conforming receptors here and here, which means that ultimately the receptors conform with the configuration of the inciting allergen. It then produces soluble receptors that bind with the soluble, with the allergen, and those are called antibodies. And if it's an IL-4 signal from the T to the B cell, it's an IgE antibody, and then we get IgE antibodies bound onto the membrane of the basophils and the mast cells. And when they get triggered again by the inciting allergen, they release their chemicals from the preformed mediators in their granules, primarily histamine, then others are synthesized. Not all antibodies are created equally. If we go through immunotherapy, we produce more IgG antibodies to block it, and as IgE antibodies go down, IgG antibodies go up, and they're protective, and therefore IgG antibodies are not really significant to actually food intolerance, whereas IgE antibodies are significant to food allergy. And there are obviously a host of other cells that are involved. And what we focus on primarily are these. And when they get activated, they look like this, and they produce chemicals like myeloperoxidase and other free radicals that cause damage. The evolution of the process, the evolution of the process left us to a method to just measure blood cells and see if they respond. And I think, and how we run our test then is to measure a sample of blood cell with an allergen versus one without, and see how it impacts the patient's cellular population. And here we see a cellular cell that's not reactive on a test, and here we see cells that are reactive on the graph, showing how they actually look under a microscope. They're getting deformed, they're dying, and they're releasing their chemicals from inside, which are toxic. And that's a more extreme example. There are lots of things that can trigger the cells. It's not just the classical allergic reaction. There are toxic mediators. Again, there are pharmacologic mediators. It has to do with our inheritance. It has to do with how healthy our gut is. It has to do with other epigenetic phenomenon. Do we smoke? Do we exercise? Do we get along well with our husband or our wife at work? All these things influence our genes and influence how we interact with the environment. So we put out a report which identifies the safe foods and identifies the non-safe foods and degrees of reactivity. We found that when we looked at these foods in different methodologies, they actually do produce free radicals. And here I come to probably the most interesting part of what all this background leads us to. And this is a quote about from a professor emeritus at the University of Los Angeles writing in his book about the biological basis of aging. And here's the connection. He says that phagocytes, which is what we've been talking about, generally generate high levels of oxygen radicals. They store in tightly sealed intracellular compartments called granules, hence the name granulocytes. This is a major source of damage in chronic inflammatory autoimmune reactions such as rheumatoid arthritis and can lead to serious tissue loss. The damage done by reactive oxygen molecules needed to operate living cells can be enormous. Any molecular species can be attacked. No molecular species is immune. Radicals can attack and deform protein molecules, which of course can lead to mutations that can lead to cancer. They can disrupt structural complexes and block the action, or she says inhibiting the action of important enzymatic functions. And they can also attack the individual nucleotide bases that make up both the nuclear and the mitochondrial DNA. Now, that is very interesting because what we find is that in people with inflammation, they have shorter DNA strands. How many of you are familiar with, have heard the expression telomeres? Can I say? Good. Telomeres are the ends of DNA that protect it. Every time the DNA gets copied to produce a new cell, there's an enzyme that must seed itself, just like a railroad car over a track. Seed itself over that end stretch of DNA, and that small part that has covered by the railroad track, by the enzyme, okay, reverse transcriptase, does not get copied. So what happens is the DNA gets shorter and shorter and shorter and shorter. At a certain time, when the DNA gets too short, the telomeres get too short, the cell senesces. It can no longer reproduce. That process is actually accelerated through inflammation, through the damage done to DNA, as indicated by our immunologist from UCLA. And older participants, younger, older but not older participants, each shortened kilobase pair of telomeres correspond with a threefold increase of myocardial infarction. Now there's a relationship there. It's not necessarily causing it, but the inflammation is probably causing it. And again, the same thing was found that an inverse relationship was seen between the length of the telomeres and important markers of aging. Glucose tolerance, diastolic blood pressure, thickness of the carotid artery, and levels of inflammatory mediators like IR6. It also causes imbalances in our hormones. We produce more cortisol to dampen the effect of inflammation, and that suppresses the generation of sex hormones. And this could be, as many people are pointing out, such as Ricker from Harvard, the cause of cardiovascular disease and all aspects of metabolic syndrome. These cardiologists have reported that the damage